Good morning, everybody, and thank you for inviting me. I spoke to this society about 15 years ago to describe the work that we're doing on fish migration, so this is going to be a bit of an update. As we've heard, animal migration has fascinated people since the beginning of time, and it's only with the advent of these modern devices that we're able now to start answering some of these ancient questions about where animals are and where they go and how they make their journeys. I'm going to talk about why we should study fish migration, how we go about following individuals, how we then take that information from following individual animals and scale up to populations, and then a little bit at the end about how we can use our understanding of migration in terms of what's happening with climate and in terms of giving advice about conservation and management of fish populations. So why is migration important to fish, firstly? As a fish develops from an egg, most fish eggs are about 1 millimeter in size. Some of the sharks are much bigger, but most bony fish have eggs of about 1 millimeter in diameter. And as they grow, they change size dramatically. And a tuna can end up as a very large adult, although it started as a very small egg, went through a larval stage. And it's not difficult to understand that the habitat requirements at different stages of the life cycle are going to change. And so if an animal moves between different habitats as its requirements change, it prospers, and so it can become abundant, and the population can become abundant. So moving between different habitats is good for the fish. Why is it good for fishermen? Because it is good for fishermen. If a species is abundant, because it is migratory, it becomes commercially attractive because they can be caught at a lower cost. There's lots of them, so you can go out and catch lots in a short period of time. The availability is more reliable, so you can build up a market which you can supply. And so, as I said, fishermen can sustain a supply. So migration is important to the fish, but it's also important to fishermen because of its consequence on fish populations. And why is it important in terms of fish biology? Because that's what I am, a fish biologist giving advice to government. It allows us to identify what a stock is. You will understand, you will know, I've heard about fish stocks. But if you catch cod in one place at one time of the year and you catch them somewhere else at another time of the year, is that one group of fish moving backwards and forwards, or is it two groups of fish moving to other areas that you don't know about? If you don't understand those movements, you don't know whether you're dealing with one stock or two stocks. So if you can't identify your stock, you can't do a stock assessment. A stock assessment is what fisheries biologists do to work out the size and the age structure of a population, which they can then use to calculate how many fish you can safely take from that stock without damaging it biologically. So if you can't identify your stock, you can't do a stock assessment, and if you can't do a stock assessment, you can't sustainably manage the fish's exploitation. So understanding fish movement is also important to fish biologists and fisheries biologists. So what do we need to know? We need to know where fish are in time and space, and we also need to know where they're not, because we need to understand that. We need to know what they're doing when they're in their various habitats, particularly in relation to what's available to them. So we need to know why fish go where they go and why they do what they do. Once we have this information, we can firstly use it to build biologically realistic predictive models which we can use to ask what-if type questions. What if this habitat changes? What are the fish likely to do in response to that? And we can use the outputs from those models to give advice to government and to fisheries managers. Or we can sometimes use that information directly, just a direct understanding of a bit of biology to give advice. Okay, so here are animals in a population, in a habitat. If we knew nothing about those animals, we might assume that they're randomly distributed. But if you know anything about animals in an environment, you know they're not randomly distributed. They're usually patchily distributed. But if we know nothing about the environment, we can't explain that patchy distribution, and so we can't start predicting what's going to happen if the environment changes. But if we know something about the habitat as well, then we can start to understand how and why the animals are distributed as they are, and then we can start making predictions. We can predict what will happen if the cafe runs out of donuts, and if the 10:25 turns up on time, we can predict what's going to happen there. And it's a similar sort of thing we're trying to do with fish in their environment. Now, Phil's just told us some of the wonderful things you can do with small tracking devices on birds. And as Bob said in his introduction, before we had all these devices, you could understand quite a lot about bird migration simply by looking at them. European swallows, you see them arriving in the spring, you see them disappearing in the autumn, so you know that they're going somewhere else. When they arrive, you can see where they're feeding, you can see where they're building their nests. If you go up and look into the nest, you can see how many young they have. So you can tell quite a lot about what's going on with the population simply by looking at them. With fish, particularly in the sea, this is our problem. You don't see an awful lot of fish. Okay? So we have to overcome that, that limitation and try and find out what they're doing in their natural habitat. The simplest method, as Bob talked to us about right at the beginning, for fish is a similar thing to ring it in the same way that you would ring a bird. This is a mark recapture experiment done by Dutch colleagues a long time ago on plaice. Plaice spawn in January and February in the North Sea, and we learn about their migrations by putting a small plastic disc, which we attach with a steel wire to the fish. You go out and you catch large numbers of fish, for instance, large numbers of plaice, for instance, on a spawning ground. You catch them in a benign sort of way so you don't damage them. You put the plastic disc on them and then you release them back into the sea. And you tell the fishermen if they catch a tagged fish and return it to us and tell us when and where they caught it, they get a reward. We know where they tagged them, the fishermen tell us when and where they caught them, and we can work out, in the same way that you do with bird ringing, you can work out approximately where these animals are going to. So these are spawning grounds for plaice in the North Sea, one off Flamborough, there's another one down in the southern bight of the North Sea, and there's one in the German bight. And this Dutch scientist did tagging experiments off Flamborough and in the German bight, catching them when they're spawning in the winter, and then releasing them. And if you look at where those tags are being caught by fishermen during the following summer, from Flamborough, they're spreading out all over that area, demarcated by that line there. In the German bight, they're spreading out over that large area, and in the southern bight, they're tending to spread out over this area with a bit of overlap. If you look at where those tagged fish, it's not the same fish because you only catch tagged fish once, but if you look at where fish that you tagged are being caught the following year, the following January, the fish that were tagged at Flamborough are being caught back at Flamborough. The fish that were tagged in the German bight are being caught back on their spawning ground in the German bight, and there are a few confused individuals in the middle. Okay, so that's what you can do with simple mark recapture experiments. But it's very coarse, and it doesn't tell us anything about what happened to the fish that didn't get caught, and it doesn't tell us anything about what the fish were doing between release and being recaught. So in the late 60s, the early 70s, my colleagues at Lowestoft took a leap forward with, you can't track marine animals with radio devices because radio waves don't go through seawater. Seawater's a conductor, so radio waves don't go through seawater. But you can use sound, you can use acoustics. And if you're familiar with what the Navy do, they use sonar which goes ping, and you wait and you time the amount of time it takes for the echo to come back, and you can work out how far away the object that was reflecting the sound was. If you have a directional sonar, not only can you work out how far away it is, that object, you can work out whether it's over there or whether it's over there. In the late 60s, we acquired a device which is on our research ship. We don't have that research ship anymore, called an imaging sonar. And what it does is it sends out a wedge of sound, and the echo is scanned very rapidly as it comes back in over an array. It's scanned 10,000 times a second. And that uses the sound to create a visual image of what's going on under the ship. The Admiralty had developed it for looking for mines and submarines, and when they'd finished developing it, they then asked us if we wanted their development device because they no longer needed it, and we acquired it. Originally, the idea was to use it to look at how trawl gear set behind fishing boats. But my colleagues there at the time who were biologists thinking, well, we could actually look at how individual fish respond to that fishing gear. Individual fish don't reflect an awful lot of sound, so on its own, it's not a lot of use. But our electronic engineers developed a little thing called a transponder. If you see the place at the top there, it's got a little sort of tube attached to it. That's an electronic transponder. And what happens is it hears the ping from the ship and it responds with a ping of its own. And that then appears as a bright spot on the screen and allows you to follow an individual from the research ship. And in the late 60s, early 70s, and through into the 80s, my colleagues did a lot of work tracking the migrations of plaice in the North Sea. And what they discovered is that their movement is aided by the water currents. This is the vertical movement of a plaice like that over the period of about 6 days. The solid line going up and down is the vertical movement of the plaice. Because the acoustic device allows us to see the seabed as well, the dashed line at the bottom is the seabed. So you can see this fish is going up and down regularly. 6 hours up, 6 hours down, 6 hours up, 6 hours down. And that vertical movement is in phase with the local tidal streams. So this fish is out here in the southern North Sea, going up and down in phase with the water currents. So it comes up when the tide is going north and it gets carried north. When the tide turns and starts to come south again, it goes to the seabed, so it doesn't get carried back south. And so it gets carried in the direction it wants to go in a series of hops, and then it goes back the other way when it's finished its migration, and when it's finished spawning and is migrating the other way. So that was a great piece of biological understanding generated by very early electronic devices that were able to put on fish. But you can imagine, can't you, that going out in a large research ship with 6 scientists and 17 crew to track one individual is a very expensive way of going around about getting biological information. And of course, we can only track one fish at a time, because you can be pretty sure if you tag 2, one will go one way and one will go the other. And you can only track individual animals for a relatively short period of time, because the device is going ping every 2 seconds and it runs out of battery. So whilst that tracking methodology told us some fascinating things about fish biology, it didn't tell us everything we needed to know to be able to build up an annual cycle of their migration routine. So we needed a new technology. And in the late 80s and the early 90s, we could take advantage of the massive advances that were taking place in microelectronics, transistors, as Bob said earlier on. And we were able to build small computers for fish. This is our first generation. It doesn't look quite so small now. It looked pretty small at the time, but they're a lot smaller now. This, for a plaice, this electronic tag has a microcomputer inside it. It has a pressure sensor, so it records depth, so we can record that vertical movement that I showed you earlier. It also has a temperature sensor. Now, we don't follow these animals. We don't have to. What we do is we put them, because these are commercial species, they get caught. We tag them with an electronic device. We tell the fishermen they'll get a nice big reward if they catch one of our tagged fish and send it back to us. And then we can download the data and analyze it and work out what the fish has been doing. And in the first generation of devices, we'd store about 32,000 data points, which meant we could record depth every 10 minutes and temperature once a day for about 8 months. That compares with being able to track an individual for about 4 or 5 days. So a massive increase in the amount of data that we were able to gather using this technology. And this is the data that we got from our very first plaice with one of those devices attached to it when it came back after about a month at sea. And what you can see is this is the fish being released. This is depth against time. The fish is released. Up the top there is the surface. It comes to the seabed, sits on the seabed for a while, and then it starts doing some vertical movements. And if we expand that bit up there at the top, you can see that's that very regular pattern of vertical movements that's linked to the tidal streams. So it was migrating there. And if we come towards the end here, that bit expanded is up there, and I've added day and night there. And what you can see is the pattern of behavior has changed. Up here, over here, the vertical movement is related to the tidal streams because the animal is moving. Over here, the vertical movement is no longer related to the tidal streams. It's related to day and night because it's reached its spawning ground. And what we were able to do, because we know these animals are moved about by the water currents, we were able to build a computer model that had a database of tidal information that could calculate the speed and direction of the tide at any point around the UK. So we can then take the vertical movement of a fish from a known starting point and calculate its spatial movement based on it being moved by the water currents. And so that's what we've done here. As the fish being released off Lowestoft, in the early part of that period at Liberty, it was going up and down in phase with the tides, so it was carried down through the southern North Sea, through the Dover Strait, into the eastern English Channel. That's where it changed its behavior and starts going up and down in phase with day and night. And day and night are out of phase with the tides, so the fish just goes round and round in circles, looking for its spawning ground and trying to find partners. This is where our model got that fish to in reconstructing the track, and this is where the French fisherman said he caught it, and obviously it fits well, so we really trust French fishermen about what they tell us. This is about the third or fourth fish that we got back of that release in 1993. It was released in a very similar position to the first one, but after this was about a month at liberty, this was 2 months at liberty, and it had only traveled about 80 kilometers to the north. So our initial assumption was this fish hadn't done very much, hadn't gone very far. But when we sort of glanced at the vertical movement that we had printed out, it was obviously doing a lot of behavior, but it didn't seem to have gone very far. So we used the same method to reconstruct its track, and this is the track that that the model suggested the fish had done. It hadn't just gone a little bit to the north. It had first gone up to Flamborough, where there's a spawning ground, but it hadn't stayed there. It had then come rapidly down south into the eastern English Channel like this fish had, which is where we think it had spawned. These are all female fish because it's only the females that were big enough to carry these tags. Males tend to be quite a bit smaller. And it was then coming back into the southern North Sea, back into its feeding ground when it was recaught. Our model got it to about here. The recaptured position was supposed to be a bit further north. So this suggested that this fish hadn't moved 80 kilometers, it had moved 800 kilometers in 2 months. And if you're thinking about managing, for instance, stocks of fish by closing areas to fishing at certain times of the year, if your mark recapture data suggests the movement isn't very much, then you might suggest you could close a small area and protect those fish. But if it's actually moving over a much bigger area, your closed area isn't going to be very effective. So it shows how, right from the beginning, this better understanding was helping us give advice about managing fisheries. But this was very unexpected. We really wouldn't have expected a fish to go first to one spawning ground, possibly spawn, probably not, and then come all the way south to another spawning ground. And some of our colleagues were a bit skeptical about taking this data, putting it into a computer model, and reconstructing a track like that. So as I said, I hope I said, there's also a temperature sensor in these devices, so we went to the temperature record. And in those days, we didn't have fancy satellite images of sea surface temperature. We had to go to the Germans and get their data. But this is what the temperature of the seabed looks like in the North Sea in this time of the year. All you need to understand from this is it's about 6, 7, 8 degrees in the North Sea, and it's about 9, 10, 11 degrees down in the eastern English Channel. And this is the temperature record of that fish. It starts at about 6 degrees, which is consistent with being released just off Lowestoft. The temperature then declines over the next 3 weeks, which is consistent with going north. It then rapidly warms to about 10 degrees, which is consistent with coming down into the eastern English Channel. And then it cools back down to about 6 or 7 degrees, which is consistent with going back up into the North Sea. And then it cools further to -4 degrees, which is consistent with being on ice in a fisherman's hold. So the temperature record, in a coarse sort of way, confirms that big geographical movement that we had developed by putting the vertical movement into a computer model. But then we noticed something which we hadn't expected, completely unexpected, is that when the fish sits on the bottom, this is just a short piece of data from one of these places. You can see the periods when the fish is up swimming in mid-water. But when it's sitting on the seabed doing nothing, because we're recording pressure, we record the rise and fall of the tide. So that gives us the time of high water and the tidal range on a particular day. And in the same way as using daylight to calculate position for animals, birds, and things that are where you can get sunlight, this gives us a way of calculating the position of the fish. Because in the North Sea, I'm not going to go into the detailed hydrography, we have what's called an amphidromic system. We have a number of amphidromic points, and those points are the points around which the energy wave creating the tides rotates. Not the water. The water goes backwards and forwards, but the energy wave rotates, which means time of high water rotates around the amphidromic point. These are hourly differences, so you get high tide here, and then an hour later here, and then here, and then here. And then tidal range, that's the depth difference between high tide and low tide, radiate out from the amphidromic point. So at the amphidromic point, there's no rise and fall of the tide. As time passes, as you come out from the amphidromic point, the tidal range gets bigger and bigger. So you can go to the track, and that's what we've done over here, find periods when the fish is sitting on the seabed, take the time of high water and the tidal range, put it into a different computer model of the tides, and it tells us whereabouts the fish is. If we go to one of these points up at the top here, the depth data is in the top corner. We can calculate the time of high water and the tidal range. Areas, you probably can't make it out, but I can't. But we can plot the areas with the same time of high water, plot areas with the same tidal range, and then plot the areas where both coincide. And what you can see is, I hope, is that at 3 points in this track where we thought the fish was from our reconstruction of its movement, the tidal data agree pretty much in these 3 positions. And at the last point here, as you can see in the depth data, when it's sitting on the seabed, there's no rise and fall because the fish is actually on the amphidromic point. And so we thought the fish from its track was just going through the Dover Strait, but it's actually further north off Lowestoft, which is close to where the fisherman said he caught it. So this was giving us a good position. So now we had 2 ways of reconstructing the movements of fish based simply on depth and temperature. Firstly, by putting their vertical movement into a model of the tides, and it would show how the animal would be moved around. But we could also use this depth data when the fish is sitting close to the seabed or on the seabed to calculate its position. And we don't need any satellites and we don't need any radio to do all of that. So making those huge advances and those successes allowed us to scale up our research and get extra funding from government and the EU. We were able to develop further types of devices, so we're taking advantage of developments in electronics. So this is the first device, 32,000 data points. You can see how big it is. The device we're using now is much smaller and recalled 1 million data points. And we're able to tag fish all over the North Sea and look at how changes in behavior relate to changes in position, changes in habitat. Okay. And now, finally, to summarize for the place, we were able to plot, we tagged, as I said in the previous slide, hundreds of fish, and look at where they are at different times of the year. They're color-coded here at the beginning for where they are on their summer feeding grounds in June through to October, and then we can follow the movements of those fish, the colors, as you go through the seasons, through the spawning cycle and back to the summer. And this is then the population understanding. This is scaling up from individuals to populations. This is summarized in this slide. So the summer distribution of these 3 populations, A, B, and C, is shown in the red colors there. And the arrows show the different spawning grounds they go to in the winter. The important thing to take from this is that the original mark recapture work done by a Dutch scientist back in the 60s suggested that these fish were on discrete spawning grounds during this winter and overlapped a bit during the summer. What this is showing us is that they're actually on discrete feeding grounds in the summer and they overlap on their spawning grounds in the winter. And that's important because one of the things we've been trying to do is use genetics to identify population structure, and there is no population structure for place in the North Sea from a genetic point of view, and the reason for that is because they're all mixing up when they're spawning. So the tracking technology confirms the genetic biology, and it allows us to work out where the populations go and how they intermix. And so we can advise fisheries scientists and managers how the impacts of varying fishing activity in different places is likely to impact on populations. Okay, now we're going to move on from the place story to some of the other species we've been working on more recently. As I said, we're trying to link fish biology and the environment, knowing where, what, and why, to generate biologically realistic predictive models to give evidence-based advice. I'm firstly going to talk very quickly about some work we've been doing with Atlantic cod in a large EU-funded project with a load of partners across Europe, working on cod. We called the Project Codacy. Don't worry, it gets worse. Thousands, I think it says somewhere here, over 3,000 tags have been deployed on these fish, giving us over 130,000 days worth of data. You can imagine how much it would cost to generate 130,000 days worth of data if you were paying a research ship £20,000 a day to steam up and down the North Sea. Much more cost effective doing it this way. Now this is apparently it's going to happen automatically. There it goes. Follow that line, the red line going across. This is a year in the life of the cod. All the little green lines are where we can get a tidal location. And this is the movement of that fish in space using the method I described before. So this is, the blue line is the depth record from the cod over a whole year. And a computer program is calculating its position based on the tidal data in that data at the point where that red line is moving through. The release position is that green triangle. The fish is recaught back up in the North Sea. But as you can see, it's spending most of its summer out here in the Western English Channel, in deep water called the Herd Deeps. And then as you get towards the end of the year, we're in November now, these fish, cod also spawn in the winter. This fish now starts moving back up the channel, back up to the position at which it was caught. So we can now use this tidal data to calculate the position of fish automatically with a computer program like that. This panel over here tells us something about cod behavior in relation to the environment. These are two female cod, one on each side. I'm going to explain what these pictures mean in a minute. One in the North Sea from the period April through to October. Same year, another cod in the Irish Sea. As I've explained, we're monitoring their vertical movement, so that can give us an index of activity, depending on how much vertical movement they're making. If they're not moving very much, we color-code it in white. If they're moving a lot, we color-code it in black. And then we plot it for every hour. The panel is 48 hours wide, 2 days, and we double plot. So we plot day 1 and day 2 and day 3 and day 4, all the way down. Okay, in the North Sea, in April, May, through into June, you can see the animal, the cod, is active most of the time because it's finished spawning and it's migrating back to its feeding ground. In June and July, it's got to its feeding ground, it comes out of the water column, goes down to the seabed, and it's just sitting very close to the seabed, not doing much, doing a bit of feeding, just not moving very far, relatively inactive. Towards the end of July into August, the cod starts to become active, but only at night. So you get activity from midnight through to dawn, inactive through the day, active through the next night, inactive through the next day. As you move through into the autumn, the period of activity gets longer because the nights are getting longer, and the intensity of activity increases because they're feeding much more actively, building a gonad. Middle of October, this day-night activity pattern ceases, and they're active all the time because they're now migrating back in the water column back to their spawning ground. Okay? That's a cod in the North Sea. In the Irish Sea, it's completely different. Same species, same size, same sex, different habitat, completely different behavior. Now on to basking sharks. This is work we did with our colleagues down at the Marine Biological Association in Plymouth. Basking shark is the second biggest fish in the world. It's not a commercial species, you're not allowed to catch them, so we can't use commercial catch data to tell us anything about what the population is doing. But the Marine Conservation Society used public sightings records to try and provide an index of population abundance. And this is what those sightings records were showing through the late 80s into the early 90s, a rapid decline, or relatively marked and rapid decline in sightings. Now, if that was catch data for cod, you'd be pretty worried about your population, what's happening to your stock if it was going down like that. So we got funding to do a project with basking sharks using tags which are very similar to the ones that you saw earlier. They record depth and temperature. They also record daylight, so we can get position from daylight. They're attached to the fish, but we can't catch the basking shark. So what happens is at a certain time, a little corrodable link will go, the tag will pop to the surface, and it will transmit the data that's been recording back to us via satellite. And from that, we can work out what the fish is doing. And this is the track of one basking shark. I'll try and explain where we are. That little black point there is the tip of Cornwall. And this is the shelf edge. So this is Western France. So the basking shark, and the color here is chlorophyll A. So it's an index of chlorophyll abundance, plankton abundance. And And what the basking shark, they're filter feeders, they feed on plankton. It was feeding off the French coast here, and it then moves all the way out to the shelf edge to feed out there. So we can relate the movement to the food resource that the animal is exploiting. And this is important because if the fish is close to the coast, it's going to get seen and sighted. If it's way out on the shelf edge, it's not going to get seen and not get sighted. So what we now know is that we can tune sighting surveys by where the food is abundant at the time of year to work out whether the fish are actually in decline or whether they're just not being seen because they're somewhere else. And if we now look at the rest of that sighting survey, after the mid-90s, the population goes back up again because the population doesn't go back up, the sightings records go back up. That couldn't be accounted for by population growth because basking sharks don't breed anything like that fast. But this sort of cycle is about an 11-year cycle, which we don't know that this is a speculation, but that's a bit like the cycle length of the North Atlantic Oscillation. So it's quite possible that there are environmental cycles driving where the plankton tends to be. When the plankton tends to be close to the coast, then we see basking sharks. When it's further offshore, we don't see basking sharks. So this information allows us to tune those sightings records. I told you our project names got worse. We started off with Codicy for cod. We then got a big project to work on eels, and not surprisingly, we called it Iliad. Eels are in massive decline. The recruitment of young glass eels now is about 10% of what it was 30 years, and we don't know what's happening and why it's happening. They have a fascinating biology, I'm sure you know, but I'm going to tell you. They're supposed to spawn in the Sargasso Sea. The eggs and larvae drift on the currents back to Europe, and they come into fresh water. They swim up rivers. They end up in rivers and lakes where they grow, feed and grow. And then at some point, they will turn into silver eels to migrate back to the Sargasso Sea. They can do that as early as 8 or 9 years old, or they can wait until they're sort of 30 or 40 or 50. So you can have eels leaving fresh water over decades at the same rate, even if your population isn't, your recruitment of glass eels is failing because you've got a huge stock of eels in fresh water. So we don't really know much about what's going on. And this is one of our most recent projects, and the satellite tags now, because these are way out in the open ocean, we can't track them. We want to use satellite tags that will pop up in the same way that they did with the basking sharks, but the ones we used on basking sharks were far too big for eels. So the technology has advanced, and they just produced some that were small enough. I'm just going to go quickly through. This is what the migration pattern is supposed to be, and it shows you the distance that the animals are supposed to migrate. And that migration pattern was determined in the 1920s by a guy called Schmidt. And in the intervening, by going out and trawling in the sea, catching larvae and plotting the size of the larvae depending on where they were. The smallest larvae were close to the Sargasso Sea, and as you move back towards Europe, the larvae are getting larger and larger and larger. So that's how they worked out that eels probably spawn in the Sargasso Sea. But nobody's ever found an eel egg in the Sargasso Sea or anywhere. Nobody's ever caught spawning eels at sea. Okay. So we don't actually know that they really do it. And things haven't improved much over the past 100 years. So we use these devices. They're still quite big, and we can only tag eels. This is the size of females and males, so we can only tag the very largest females. To tag 22 eels, we had to go through 10 tons of eels, live eels, to find the large ones. And this is how that technology works, and it's going to start automatically, I hope. Here it comes. So there's the tag popping off. Pops to the surface. Starts transmitting. And that data is then picked up by a satellite and transmitted back to us. So we can use this information to work out what the eel's doing. Because that technology was so large, and we could only tag the largest of eels, we took another approach to run alongside it, using the sorts of tags that we've been putting on cod. But with the cod, we rely on those fish being recaught and the tag found and sent back to us. As I said, that's not going to happen with the eels because they're way off in the Sargasso Sea. So we had the idea of having a message in a bottle. We took our little tag and we added some floats to it. The floats in this case are in a sausage of floats all connected because an eel is long and thin, and so we have a tag that's long and thin and it goes inside the eel. And so in addition to those satellite tags, some of the eels were tagged with these devices. The idea is they go out, that we tag them internally, they migrate out, they spawn, they die, they disintegrate, the tag floats to the sea surface and drifts back, and people find them on beaches and send them back to us. And this is the sort of thing that we discovered eels do. This is an eel escaping from the coast, gets out into the Atlantic, and it starts going up and down in phase with day and night. And if you follow them further, they carry on doing that, getting deeper and deeper and deeper as they migrate out towards the Sargasso Sea. This is showing you the depth, so they're going down to about 600-700 meters during the day, we think, to avoid predation, and then coming up to about 200 or 300 meters at night. And this is a summary of the data that we've got so far, giving you the distance and the speed and the bearing of these animals. The release sites are in green, either off France or off Galway, and the pop-up positions are in red. And what it seems is that these animals are seen to be making to the Azores, which is a little south of where they should be if they're going all the way over to the Sargasso, which is over here. And we haven't had anything back further west than the Azores yet. So either there's a lot of tags out there waiting to come home, or the Azores is something rather important in eel life history that we didn't know about. And finally, this is an eel data set that we couldn't understand. It's released in Galway. Up here, this is over a few days. It's going up and down in phase with day and night, going out through Galway Bay. And it comes out into the Atlantic, but it doesn't go up and down in phase with day and night. It's doing something different, and then the tag pops to the surface. That's the depth record. And we thought, what does this mean? We then looked at the temperature record. And that's the same record, but now coded for temperature. There the eel is in Galway Bay at about 9-10 degrees cold, comes out into the Atlantic, and suddenly the temperature goes up, and this is in December. The temperature goes up to about 20 degrees, and it comes up to about 25 degrees, and then cools down a bit, and then pops to the surface. 25 degrees in the Atlantic in December? No, because this is not an eel. This is a shark. Because this eel was eaten by a shark. And large laminid sharks keep their stomachs warm to aid their digestion. So although we think they make vertical movements to avoid predation, it doesn't always work. Thank you.